Okay, so we're looking at an example of the kind of data we're going to collect based on comparing the habitats, and we can see there's one row for every observation. So here we have a sample that was taken from a rock location. It's numbered as replicate 1, and there's an indicator as to whether or not we found various examples of different species within that particular sample. And this is summarized as a species richness. So we've reduced information on the kinds of species that are available to a single measure of species richness within that particular sample location. And we have a number of replicates from different rock sites and a number of replicates from different pool sites. So the first thing we might want to do is just do some visual checking on our data. So we have a graph menu that makes various kinds of graphs available. The two kinds of graphs that we might be interested for these data, possibly the individual value plot. So if we open up the dialog box for individual value plot, we're interested in grouped data, and the variable we're going to graph is the species richness indicator. So we select that for the graph variable box, and then in the categorical variable box we want to select the habitat. Various other options here let us set the title and the axes labels for this graph. Trying to keep the video short, won't go through all of that, but they're all there. We'll just have a look at the plot, and this gives us a plot which tells us, for instance, in the pool habitat there were 2 samples where the species richness was 6, whereas in the rock plot there were also 2 samples where the species richness was 6. With slightly more data than this, it would make sense to look at a box plot. So as a reminder as to how we do this, let's go to the graph and select a box plot, and again we're looking at grouped data, and again we're looking at putting the species richness in the graph variable box, and we're looking at putting the habitat in the categorical variable box. Again, we should select the labels and axes options appropriately, but if we just have a look at the visual, we get a box plot of species richness. What we're most interested in is convincing ourselves that the variance at each habitat is the same. We can be a little bit more formal there and actually calculate the group summary statistics. So if we go to stat, enter basic statistics and ask to see the display of descriptive statistics, variable we're interested in again is the species richness, and the optional by variables that we're interested in is to see how this varies by habitat. And if we select this, the procedure will print out some information. So we have 20 samples taken from each habitat. The mean species richness within the pools was 7.95. Within the rock habitat it was 4.6. And if we check over on the variance, we actually get given the standard deviation. It's 1.5 in the pool habitat and 1.1 in the rock habitat. And we now have to make a decision as to whether these numbers are close enough to consider that both sets of samples were ultimately taken from a population with the same variance or not. On the basis of such a small sample size, it's difficult to really tell. There's no clear evidence that they're different, so we'll carry our analysis on with the assumption that they do indeed have the same variance. And what we wish to do next is a formal hypothesis test. So the null hypothesis is that both populations, both sets of samples were drawn from a population with the same mean. We're hoping to reject that null hypothesis. And rightly or wrongly, the go-to test in that situation is to do a t-test. So if we go to the stat menu, this is offered under the basic statistics menu, and we're looking for something called the two-sample t-test. So we select the two-sample t-test. Essentially, it's the same menu as we've had before, only the terminology is different. In the samples box, we're actually asking for the variable of interest, so that would be species richness. And in the sample ID box, this is where we tell it there's two different groups of habitat. They're denoted by the variable called habitat. And if we select OK, the numbers will get crunched and it will carry out a two-sample t-test. And it's telling us here, what am I doing? I'm doing a t-test for the difference between the population mean of data taken from the pools and the population mean of data taken from the rocks. And under the null hypothesis, if these populations had the same mean, the mean of the first population minus the mean of the second population would be 0. It then tells us that the sample means actually had a difference of 3.3. And if we leap a line, it goes off and does a t-test. It tells us that the t statistic we obtain from doing this test is 7.72. So this is a measure of the difference between means that's standardized for the standard errors of the mean. And the probability of obtaining such a large t-value, if the null hypothesis were true, is given by the p-value. And this, as we can see, is very, very small. And by convention, if this p-value is less than 0.05, we regard this t-value as too big to be consistent with the null hypothesis, so we reject the null hypothesis. So we reject the null hypothesis that the two population means are the same, and we make a decision that the evidence in our sample entitles us to claim that the population means are not the same. In other words, we think there is a difference in the population means of species richness found at pool sites when we compare them with the species richness found at rock sites. We get much more interesting scientific information from looking at the confidence interval, and we're told that the 95% confidence interval for the difference is 2.43 and 4.16. This is telling us if we repeated this kind of survey infinite times, whatever the true difference is between species richness at pool and rock sites, 95% of the time we would find a difference that lies somewhere in the range 2.4 to 4.16. So this is useful in that, as well as telling us there is a difference, it's an attempt to quantify how big that difference might be.